Hello and welcome to the next lesson for higher biology. This one is looking at chromosome mutations. So previously we've been looking at gene mutations and what happens when a single nucleotide in the DNA sequence can be changed. This time around we're going to look at chromosome mutations before we move on, and next time we'll be looking at evolution and how all of those mutations can have an impact on new species being formed. So what you need to be able to do for this one is to describe and draw the 4 types of chromosome mutation, and you need to be able to explain the importance of duplication as a mutation to evolution. So chromosome mutations come in 2 main forms. You can either have the structural modification of a chromosome, so one chromosome is changed or modified somehow, or the number of chromosomes present in the cell is changed. When we talk about chromosome mutations, it's important to remember that we're talking about mutations that are going to occur during the mitosis phase of new cells being produced when we're looking at sperm and egg cells. If you have a mutation occurring randomly in any regular body cell, that's actually not going to have too much of an effect on the organism. Because it's only affecting one cell, the rest of the correct chromosomes in the rest of the cells are going to be able to take over and manage within that organism. So mutations really only have an impact when they occur during the phase of sperm and egg cells being produced. So as DNA replication is occurring for that process to happen, there may be a mutation that occurs, and in this case it may be a structural mutation, or it might be a change to the correct number of chromosome mutations. Therefore, when that sperm or egg fertilises or is fertilised, that mutation will then be duplicated for the rest of that embryo that's formed. So mutation will be copied and copied and copied and will soon be present in every single cell, and that's when it's going to have the impact. So the first type of mutation that we're going to talk about is actually a structural modification. If this is our chromosome here, and I've made it super simple, we've got one chromosome, each box with a letter in it is really just indicating a specific gene. So gene A would code for protein A, gene B would code for protein B, and so on. Obviously, if this was a real chromosome, there wouldn't just be a few genes present, and we wouldn't just have the 9, there would be thousands of genes along the length of a chromosome. So in a deletion mutation, what happens is there are 2 breakpoints that occur along the chromosome. After those breakpoints, the sections of chromosome break apart, and this leaves us with a section removed from the chromosome. This section and this section are going to join back together, and as a result we have lost this central section of the chromosome, so we've lost these genes here. And as I say, normally in a real chromosome this could be thousands of genes. Now it's worth being aware that a deletion could occur at any point in the chromosome. We've done it in the middle, so we've got 2 breakpoints. It could have been at this end, we could have had 1 breakpoint and lost A and B, it could have been at that end, we could have lost I, and so on. There's a variety of different ways that you could have a deletion. And in fact the example we're going to look at is this one here, where in Cri-du-Chat syndrome we have chromosome 5, you've got a deleted region. And this is what I was meaning about it having an impact after mitosis occurring for the sperm and egg to be produced. So if we were to look at a pair of chromosome 5s here, this one is absolutely perfectly regular. It may have come from either the mother or father. The other chromosome, which has come from the other parent, has a deleted region. So this has occurred as the sperm or the egg is being produced, and that's been replicated through all of the chromosomes in all of the cells. The results of this particular deletion, it is compatible with life. Not all deletions are. So this individual has been born, and we tend to notice a few things about them. They tend to have a larger head than would be standard for their body size, and they have quite widely spaced apart eyes, and they can develop learning difficulties later on in life. The next mutation we're going to look at is one which is referred to as an inversion. So again, we're starting with our very simple basic chromosomes. We have our 2 breakpoints. This time though, instead of losing our middle section of the chromosome, it gets inverted, so it turns itself round and we wind up with the genes in a different order. Now, this doesn't actually have quite as significant an impact as the deletion mutations, mostly because we've kept all of our genes. We've managed to maintain those 3 genes. They're just in a slightly different order. Now, that may have an impact if we're looking at an effect on a splice site that would normally have occurred in here somewhere, or we're changing the DNA sequence. Going from F into G would be our normal, whereas now we're going from D into G. So there may be an effect on there. However, gene E is really unaffected entirely. It's only the 2 at either end. Again, worth remembering that an inversion could be occurring at the end, so A and B could have been cut off and flipped around, and it could have gone B, A, C, or it could have been at the other end. It could include 4 sections of gene, it could be 2, it could be 1. There are a lot of different options in terms of an inversion mutation with varying degrees of impact. The next type of mutation we need to consider is a translocation mutation. In this one, it's going to affect 2 different chromosomes. So we've got our chromosome 1 and chromosome 2 here. And again, we have some breakpoints. In this case, chromosome 2, we've had a break at one end of it. And all that's going to happen is that section that has broken off from chromosome 2 is going to come over and join on with chromosome 1. So we've got these translocated genes over here. Again, this wouldn't necessarily be a huge impact because, again, we've kept the right number of genes. However, that means that chromosome 1 is now a different shape to its homologous partner. So when it's due for the time for the chromosomes to pair up during mitosis, that's unable to happen because this chromosome 1, which may have come from the mother or come from the father, can't match up with the original chromosome 1 that would have come from the other parent. So over mitosis, that's not going to happen, so cell division doesn't happen. And as a result of that, most large translocation mutations aren't compatible with life. If an egg with this particular translocation is fertilised by a sperm with normal chromosome 1s, it's impossible for the chromosomes to match up during mitosis. So no cell division could happen. So the cell's only going to ever be one size, it's only going to be one fertilised egg cell large. It can't divide any further than that. You can have what we call reciprocal translocations though. And when this occurs, you may wind up with 2 different chromosomes and they're essentially swapping different sections. So if this is our chromosome 4 and our chromosome 20, our reciprocal translocation has swapped we've got our section of chromosome 20 has come over and is now on chromosome 4, and the bottom section of chromosome 4 is now on chromosome 20 instead. Non-reciprocal translocation is the other kind, and that's again we looked at to begin with. It's when we've got one section of chromosome 4 has come over and inserted itself into the middle of chromosome 20. So again, that's quite different. We've not swapped them, and this one has just lost a piece and this one has gained a piece. The last type we need to consider is when we have duplication of chromosomal material. So if you look at our original chromosome and then our converted one, we've had a whole extra section. So we've got whole 3 new genes have been added into this. So we've got a duplicated area of the chromosome. Again, the impact of this is actually mixed depending on the types of genes which have been duplicated. So it could be a really significant impact, or it may be of no impact at all. When duplication occurs though, it can be quite important in terms of evolution. If this was our gene and it was duplicated like we just saw previously, so if we've got a duplicated section here and that's one gene, so we've got 2 copies now of gene A. So if that's our copy of gene A, that's our normal one, and this is our extra copy, it's possible for mutations to occur on the extra one. And those mutations may not have the impact that they normally would on the organism, because those little gene mutations that occur, it almost doesn't matter that they're happening because we've still got the normal gene A to be able to produce the protein that it's supposed to be producing. And these mutations, on the other hand, may be useful for the organism, so they may be beneficial mutations, or they may be detrimental mutations. If they're detrimental, then they're not going to carry on. There's going to be no benefit to the organism, but the organism won't necessarily notice any particular change because we've still got our normal gene A over here doing the work and producing the correct protein. If, however, the mutations occurring on a duplicated gene A are of use, then they're going to survive to be passed on throughout the generations, and over time we're going to be able to mutate entirely. Gene A will eventually look completely different from our original, and that's one of the ways that we can carry out mutation without actually harming our organisms to begin with. So you should be able to describe and draw the 4 different types of chromosome mutation, those being deletion, inversion, duplication and translocation, and explain that the importance of duplication to evolution is that it provides additional genes for mutations to be able to act on without actually causing any harm to the organism.